This is Tech Tuesday, where we shine the spotlight on a trending theme in the world of technology and gain a better understanding of where trends are leading in the world of tech investment. This is Tech Tuesday, presented by the Canadian Securities Exchange. Thank you to everyone for joining us live today for this week's edition of Tech Tuesday. Today's session, Precision Medicine in Cancer, presented by the Canadian Securities Exchange, the exchange for entrepreneurs and the world's fastest growing small and mid-cap exchange for the past 15 years. This session is sponsored by US Angels and John Ricci. John is founder and managing director of US Angels, an angel investor group based in Silicon Valley, and director portfolio management at Keotech Technology and Life Science, an investment firm with over 75 active portfolio companies. Today, we are pleased to have three companies joining us. Nicole Ruggiero, CEO of EpiVax Therapeutics, Len Pagliaro, PhD, CEO of Siva Therapeutics, and Dr. Jeremy Chrisp, CEO of EpiAxis Therapeutics. Mark Francis is this week's moderator, and this represents today's lineup. Our objective in running Tech Tuesday is twofold. First, it's for companies to gain visibility and traction on an accessible platform. The second is to inform capital markets players and investors about the types of companies in the tech space, while also providing new knowledge of what is happening in the world of precision medicine in cancer. For you, the attendees, we do ask that you share your ideas, leads, possible funding, and feedback with our presenters. You may even have a potential strategic relationship, and you can find their information in the profile box. With respect, we might refrain from giving management advice unless we are truly experts in the company's particular field and don't treat our presenting companies as marketing targets for services. Some housekeeping matters. If images aren't clear or pictures are incomplete, check your bandwidth, refresh your screen, call your IT, or try a different platform, as we are broadcasting on YouTube, LinkedIn, and AirMeet. This presentation is for information purposes only and is not a solicitation to make an investment in either shares or debt or to buy and sell stock. We make no representation about these companies, and if you are interested in their investor pitch deck, please connect with the companies directly in order to get detailed information. Each company's presentation will be about 5 minutes. Afterwards, there'll be a Q&A with Mark Francis. Now, our first presenter, Nicole Ruggiero, is Chief Executive Officer at EpiVax Therapeutics. She has more than 20 years of pharmaceutical and healthcare experience with a diverse background in research and development, operations, manufacturing, strategic planning, and business development. She began her career in vaccines and biologics, as well as a researcher at Novartis and later GSK, where she played a key role in the development of several vaccine candidates. Nicole has since held leadership roles in the industry in business development, program management, and operations.
Hello, thank you everyone. My name is Nicole Ruggiero. I'm the CEO of EpiVax Therapeutics. We're a US-based biotech company developing next-generation precision cancer therapies. So cancer therapy is not a one-size-fits-all because every tumor is different and every patient has a unique immune system. Hence, personalized therapies like CAR Ts, for example, are highly effective, although they are quite expensive and limited to liquid tumors only. Non-customized therapies like checkpoint inhibitors have lower efficacy, and they also come with some severe side effects and sometimes permanent. At EpiVax Therapeutics, we provide personalized cancer immune therapies that are safe, fast, and can be prepared on demand for any solid tumor. We give the patients the key to fight their cancer by providing a therapy that activates a complete immune response. We deliver the personalized therapies at record speed, providing maximum benefit to these patients. We've completed preclinical work, and we're ready to file IND and start a clinical trial. So our technology is trade secret and IP protected. The cancer immune therapy market is indeed growing fast and anticipated to reach over $300 billion in the next few years. We can design a therapy for any solid tumor, as I mentioned, and given the low risk of adverse events that our therapy provides, we are compatible with any other complementary treatments. Our initial application is in bladder cancer, which is a major unmet medical need with a high mortality rate and is inadequately addressed by the existing treatments. So what is the process for this customized therapy? Well, we sequence the patient's tumor biopsy and feed that information into our proprietary platform we call Answer to design a highly effective, personalized cancer treatment that is formulated as peptides with an adjuvant that mounts a robust immune response against the tumor. We can deliver this treatment in as little as 4 weeks, enabling us to change the paradigm and treat patients early on when they're healthier and the immune system is less compromised. So how does it work? There is a balance in the immune system where some cells promote an immune response while others suppress the immune response. When developing cancer therapies, we want to make sure that we only activate the right cells, the ones that will promote an immune response. Others in the field are indiscriminately activating T cells, those that promote and those that inhibit the immune response, which is in the end inefficient and leads to reduced efficacy. This is where we really differ from the competition. We have developed an automated platform shown here on the right that can repeatedly and effectively design therapies that activate a complete immune response. We are the only company that are able to filter out the suppressive immune sequences, therefore promoting superior efficacy with an added degree of safety. So here's a look at some preclinical animal data. The graph on the left, again, emphasizes the importance of filtering out those sequences that suppress the immune response, showing here a 5-fold difference in that response that our therapy generates compared to previous therapies or those that the competitors can achieve. Here on the right-hand side, these are results from an efficacy study showing tumor inhibition in a mouse model. Here we show that as a standalone therapy, we are comparable to the standard checkpoint inhibition therapy. But importantly, we also see a very strong synergy when we administer our therapy in combination with the checkpoint inhibitor, achieving here greater than a 90% tumor inhibition. We have an incredible team driving these efforts. Myself, I come from GSK and Novartis, and I've spent a lot of time in product development and business development in various companies. Our co-founder and CSO, Dr. Annie DeGroot, she's an expert immunologist. She is internationally known for her research in therapeutics and has over 30 years of experience in the field. The chairman of our board is a biotech leader who has co-founded several companies that have achieved highly successful exits. That includes Biogen and Protein Sciences. And then our team, last and not least, the team of our talented experts includes an awarded oncologist who is an advisor to many large pharma and biotech, and that's Dr. Steinberg. We are seeking $1.6 million to enable IND filing, followed by a Series A of $30 million to complete a phase 1 clinical trial in bladder cancer. And here on the left-hand side, you can see some relevant exits and financial transactions in the near past. I want to thank you for this opportunity to present to you today.
Next up, we have Len Pagliaro. He is the CEO and co-founder of Siva Therapeutics, where he recruited a management team, assembled a strong growing IP portfolio, raised founding capital from private investors and grants, and led the team that has generated very strong preclinical safety and efficacy data. Len has over 24 years of experience with successful commercialization of biotechnology products, services, and technology licensing.
Thank you very much, Barrington. I'm Len Pagliaro, CEO of Siva Therapeutics. We're a preclinical stage medical device company developing a technology for targeting gentle therapeutic heat to solid tumors. There are technologies for targeting heat to tumors, but they're virtually all for ablation, very high temperatures that cook tissue indiscriminately, and they require specialized, not commonly available equipment such as this. In contrast, Siva's approach is going to use simple, affordable instrumentation that fits into existing clinical workflow. This is our solution for rectal cancer, our initial therapeutic target, using a flexible sigmoidoscope that's modified slightly, but an instrument that docs are very familiar with, and also an instrument that's very affordable. Now, there are 4 things that gentle therapeutic heat does that we know about. It stimulates the immune system. It takes out cancer stem cells, which tend to be drug resistant. It increases perfusion to the tumor so that drugs can do their job better access all tumor compartments. And then finally, it shrinks tumors directly because tumors have different metabolism from non-tumor cells. It's important to point out that we're talking about a temperature of 44 Celsius or 111 Fahrenheit. And so that's higher than a high fever, but it's not ablation. Maintaining this gentle temperature turns out to be more difficult than establishing and maintaining ablation. When we talk about the distinction, ablation is, if you think about cooking steak, it's basically well done, medium well to well done, whereas hyperthermia on this chart is not even what's called blue rare. So yes, they're both heat, but they do very, very different things. We don't want to kill the tissue indiscriminately. We want to warm it up to the temperature at which you get these effects selectively in cancer cells. The reason it does that is that healthy cells are more resistant to heat stress than cancer cells. Cancer cells tend to have a higher expression level of heat shock proteins. And so heat, gentle heat, puts them into a cycle of apoptosis and necrosis, and you can selectively kill cancer cells. Now, the way we do this is with a two-component medical device system. It does involve an injectable, but there are, I'll call them precedents for that. They're not predicates in the FDA sense and that they're not to market yet, but there are precedents for injectable nanomaterials that are medical devices. So the first part is what we call Siva rods. They're precision nanorods. They're metallic gold, polymer coated. We've scaled manufacturing, very safe, excreted in the urine and the feces. The second component is a light engine. It's an infrared light engine. So it's not radiation, infrared light, basically a fancy heat lamp. The nanorods absorb infrared light, and then they re-emit it very, very efficiently as heat. We call the combination targeted hyperthermia. We have an issued patent and another patent in development around the light device and trade secrets around manufacturing, scaling manufacturing of the nanorods, which is critical. Now, there are two main challenges that this approach solves. The first is targeting heat to the tumor. Ideally, you want to heat the tumor and only the tumor. The second is achieving this level of hyperthermia instead of ablation. So the way the nanorods concentrate in tumors is because tumors have leaky vasculature. It varies from tumor type to tumor type. We've selected tumor types, and especially rectal cancer, that have a high degree of abnormal spongy vasculature. So 12 to 18 hours after injection, we see a 7-fold concentration of nanorods in tumor tissue versus non-tumor tissue. That plus regional administration of infrared light enable us to target the heat to the tumor or tumors that we want. Now, the conventional technologies do what we call outside-in heating. They're directing energy in from the outside. With our approach, what we see is inside-out heating, and that's ideally what we want to do. The infrared light travels through 2.5 centimeters, about an inch of tissue, with enough energy to excite the tissue to therapeutic temperature. And so we can get almost anywhere in the body using a flexible endoscope. And that, of course, is the approach we use for rectal cancer. We have a lot of preclinical data. Here is just an example of a mouse study. You can see large tumors on the left. Tumors are mostly gone in the middle. On the right, same animal, 59 days out. We carried this particular study out to 95 days. Excellent results. 66 of the animals survived to 95 days. In contrast, with the drug of choice for this tumor model, all the animals were down by 28 days. Our initial market, as I mentioned, is rectal cancer. So if we think about this as a subset of colorectal cancer, which is a very large market, and then if we look at our obtainable market, which we project to be a little over $2 billion, it's a substantial market, and it's an ideal market in terms of the tumor biology, in terms of integration into clinical workflow, and because surgeons are reluctant to do surgery for rectal cancer because of the devastating effects on lifestyle. Here's what we envision the patient experience to be. Patient goes into the clinic one day, they get a systemic injection, intravenous injection of Siva rods, they circulate through the body, they concentrate in tumors, they come back 12 to 18 hours later, and they get treated with infrared light through a flexible sigmoidoscope. Here's our roadmap. We're currently raising a $2.5 million round, the overall goal of which is to complete large animal studies, which will prepare us to enter the clinic. We do anticipate, because of an excellent safety profile, being on the de novo pathway. That means we will have a human as medical device, a human pilot and feasibility study of about 35 patients, and a human pivotal study of about 100 patients. So all in about $30 million, a little over, to get to market, which is very reasonable for a platform technology for cancer. Can't apply to all cancers, but it applies to a number of different cancers. Up to now, we've raised just under $1 million in equity and over $2 million in grants. We've got a strong team. On the left is our core team. I have experience initially as an academician, moved to industry in the mid-90s, been through 4 previous companies, 3 of which have successfully brought products to market, 2 nice exits out of that. Colin runs day-to-day operations R&D. He has a background in both science and in business. And my co-founder, Steve, is an attorney by training, but he's actually been co-founder of 2 previous medical device companies. We've all been part of companies that have brought products to market successfully before, which I think is really important for that kind of a track record. On the right, we have a growing list of advisors who are very strong in the medical device space and in the cancer space. Won't go through them in detail. Happy to discuss that offline, but it's a great group of advisors, and we're growing that as we speak. So we're raising $2.5 million, as I mentioned. The overall objective is complete large animal studies and file for an IDE as a device, again, in investigational device exemption. We do have a $500,000 commitment from a strategic partner, working hard to close it this quarter. So I'll stop there, and thank you very much. Appreciate the opportunity, and I think it's a great forum on precision cancer treatment.
Last but not least, Dr. Jeremy Chrisp is a biotechnology entrepreneur with extensive experience in the commercialization of Australian technology. His focus is on business development and the management of early-stage life science opportunities. Jeremy holds a PhD in physiology from the University of Canterbury, New Zealand, and is a fellow of the Australian Institute of Company Directors, AICD. He has over 30 years' experience in the Australian healthcare market, has worked with leading global multinational companies, including Merck & Company, Stryker, Becton, Dixon, and Medtronic. Jeremy, take it away.
Barrington, thank you very much, and it's certainly my pleasure to present on Tech Tuesday. I'm the founding CEO of EpiAxis Therapeutics, and it's my pleasure to present on behalf of the team to tell you the story about overcoming treatment resistance and preventing cancer recurrence. Next slide, please. Allow me to start by giving a little bit of background and some highlights on EpiAxis. We're basically a young company, started in 2015. We're private, unlisted. We're in the epigenetic space, with LSD1 being our lead program. It's not the only program, but it's a lead program. We're at the preclinical stage, and we're currently seeking to raise $12 million to get us into the clinic, and I'll talk a little bit about that in the presentation. But we've run always a very lean operation here and have outsourced to leading CROs around the world. We've got extensive intellectual property, which I'll comment on, a really strong board, and I'd like to introduce them in a moment. And we've got a lot of collaborations, including one in Toronto, which I'd like to comment on. Allow me to introduce the team. It's led by our executive chair, Dr. David Fuller. David is a physician and pharmacologist. So collectively, we have a team here of 4 MDs, 4 PhDs with over 270 cancer trials to their credit, and 11 drugs collectively have been brought to market from this group with approvals either in FDA or EMA. And we're on 4 countries at the moment. We've got the team partly in the US, partly in Italy, partly in Switzerland, and the bulk of it is in Sydney, Australia. Our focus is on LSD1, lysine-specific demethylase. We believe this is the pivotal enzyme controlling demethylation. And indeed, when you look at the Human Protein Atlas data, there you can see that it's significantly overexpressed in a vast number of solid tissue and hematological malignancies. And our focus has been very much around the breast and glioma indications. Of course, LSD1, we believe, equates to poor prognosis, and as alluded to earlier, with the use of chemotherapy and immunotherapy, you leave behind tumors that are enriched with LSD1. So we believe this is the key enzyme that's controlling aggressiveness in cancer cells. This is how we disrupt LSD1. We learned that LSD1 enters the nucleus, if you look on the left-hand side there, by binding on the shuttle proteins, important alpha. And once in the nucleus, LSD1 combines with CoREST to perform its demethylation function. So what EpiAxis does is we bind with our first-in-class therapeutics on important alpha to stop LSD1 entering the nucleus. And then we bind on the outer rim of LSD1 to stop the alignment with co-rest. So we have a very effective way of lowering nuclear LSD1. And indeed, we do this both on the tumor side of it and on the immune side of it. And this is why we've got broad applicability across multiple tumor types. So we're first in class because, as I alluded to, we're the only company that has adopted this NLS strategy to block nuclear entry of LSD1. And we think this is incredibly important and a major point of differentiation from the competitors that are out there. A quick look at our data sets. We have data on the cell lines. Most of them are around breast cancers with some focus clearly on melanomas and colon cancers. Our animal models are in the syngeneic and on the nude mouse models. But we've also been treating a number of patients ex vivo by harvesting their circulating tumor cells and immune cells, particularly from patients with metastatic breast cancer and also a cohort that have been immunotherapy resistant from melanoma. And we've also run a pioneering phase 1 biomarker study using the first time ever a combination strategy of an LSD1 inhibitor in combination with standard of care for metastatic breast cancer, nab-paclitaxel. And indeed, that showed that we could reverse the aggressiveness with those markers, LSD1, EGFR, FOXQ1 in snail, in that cohort of patients. And this was published early this year in Frontiers of Oncology. As alluded to, we have extensive intellectual property, all owned by EpiAxis. It covers 11 patent families, and we've been prosecuting them in 8 jurisdictions, including Canada. 14 thus far have been granted, including a pivotal one from the US Patent Office. This is giving us class protection for the use of all LSD1 in combination with immunotherapy, chemotherapy, radiotherapy, hormonal therapy, and with surgery. And indeed, we've been following both the composition of matter and methods of use strategy, with 7 of those directed at LSD1 and 4 being directed on our PKC theta a target as well. Just like to share with you some of our collaborations. We value these, led by Peptologics and Pittsburgh-based. We believe that they are a leading AI company for peptide drug development. And of course, in Toronto, we've got a very good collaboration with Dr. Hansen He's lab there on prostate cancer, and this is also in collaboration with the University of Massachusetts. And in New York, breast cancer collaboration there on our PKC theta programs. Just like to share with you the fact that epigenetics has attracted substantial interest from pharma. 5 recent transactions valuing about $4 billion, which shows the value that pharma now places on epigenetic therapies going forward. So in summary, we have a novel epigenetic therapy here that's really been designed to overcome treatment resistance and prevent cancer recurrence. This is because we're directing epigenetics towards both the immune side of it and on the cancer side. We've got first-in-class assets, the only company with these nuclear translocation inhibitors against LSD1. And we're seeking that funding to allow us to complete our IND to complete our phase 1 program starting in CY24. We're positioning this for standard of care to be added to standard of care, and that's the important point. We've got very strong intellectual property, and we've got a team that have got an enviable track record for prosecution. So we thank you for your interest. We believe the future of cancer therapy is here, and I look forward to today's discussion. Thank you.
Thank you to the 3 of you for presenting, Nicole, Len, and Jeremy. I chose you from dozens of presentations because I felt that you'd be a good cross-section of some of the innovation that's going on, and you're plainly leading as well. But a lot of our listeners are actually fairly much lay people. So let's start with a little bit of explanation from each of you, Nicole, Len, and Jeremy. Why don't T cells identify cancer as the enemy? What is going on there? Why don't cancer cells deal well with heat? And what is ablation? And finally, Jeremy, what are epigenetics and demethylation? If you could help us with that, that would be great.
Sure, I will. I can answer about why the T cells don't identify cancer as the enemy. So what we're really talking about here is basically how do killer T cells fail to attack the tumor, because that is their job. And this is really a result of the tumor using our own immune system against us. So the tumor itself can activate multiple immune inhibitory pathways to prevent the effective recognition of the cancer cells by these effector or killer T cells. One of these pathways involves activating what I spoke about, those regulatory T cells, which can prevent the activation of the other immune cells, such as killer T cells, hence allowing the tumor to escape. So our therapy selects for the activation of the very specific immune activating T cells, and we leave out those regulatory T cells. So that is how we improve upon that.
Should I take a crack then? So the reason cancer cells do respond to heat more than non-cancer cells is that in general, cancer cells tend to have a high expression level of what are called heat shock proteins. Heat shock proteins are well known, well characterized. And among other things, because of this higher expression level in cancer cells, they're one of the targets that's been identified, and there have been attempts to drug heat shock proteins. I'm not aware of any successes there yet, but there may well be. Our approach is a little bit different. We're saying heat shock proteins are already at a high level. What if we push them a little bit higher? Because when they do get to high levels, the heat shock proteins, some of them specifically HSP70, HSP90, can put cells into a cycle of apoptosis and necrosis. So essentially, we can push cancer cells into what's called programmed cell death fairly selectively. And it's that selective mechanism that's really valuable to distinguish cancer tissue from healthy tissue.
So, Mark, let me jump in and try and answer the question about methylation. And methylation is a chemical mark. And epigenetics uses these little chemical marks to change how the DNA code is read. It never changes the underlying code. And it's like the dimmer switch at home. You dial up or you dial down the intensity of those genes. So the wiring stays the same. And in cancer, the balance of how this is read gets changed. So what we're seeking to do is try and restore that balance by controlling that dimmer switch. And methylation is one of the little chemical marks that we use to achieve that goal.
Excellent. Thank you very much. That helps me, and I'm sure it'll help a number of our viewers. So the next thing I want to find out from you is the aha moment that occurred for each of you. And what did you do immediately after that aha moment? So, and let's go with Len and Jeremy and then Nicole.
Okay, well, that's a great question. So first of all, we did not discover the concept or invent the concept of using heat to target cancer. The concept's been around for about 130 years. What we did, though, was we started to look at what the technologies were at the time for targeting heat and what temperatures are used. And very quickly, it became evident to us that using high temperatures, using ablation works, but it's non-selective. And not only is it non-selective, you don't get any of the advantages that you get with hyperthermia. And this is things like stimulating the immune system, taking out cancer stem cells, shrinking tumors, and so on. And so we said, we've got to get to this intermediate temperature, this more gentle hyperthermia. And it turns out that that's a lot harder to do than it is to basically cook tissue. There are technologies for cooking tissue, radio frequency, microwave, and so on. And so we realized that using nanoparticles that could infiltrate the tumor and could absorb infrared light and convert that light into heat very efficiently was really a good way to go. I would say the aha moment, to answer the question, was being able to manufacture those particles. There are a lot of publications on nanoparticles, a lot of publications on nanorods. But to our knowledge, nobody has scaled nanorods with anywhere near the uniformity that we have. And so scaling manufacturing is really, really critical to doing this.
So, Mark, let me chime in on our sort of aha moment. And I think that really goes right to the heart of it. When we funded the initial animal studies and when they came back, it taught us that a combination strategy of a LSD1 inhibitor with standard of care in the animal model stopped tumor growth, whereas the standard of care did not. and the LSD1 inhibitor by itself did not. It was the combination. And that really set us off from day one, understanding epigenetics in a very different way, that it was a combination strategy. And that is why we went straight to the clinic, even without a compound ourselves, to prove this thesis that the combination of an LSD1 inhibitor working in tandem with standard of care worked. And so we went to the clinic first before having our first-in-class compounds, which is now why we're going back to the clinic. So that was really the aha moment, with the recognition that monotherapy for active disease with a LSD1 inhibitor and an epigenetic strategy in general probably wasn't the smartest approach. And this is why having that broad applicability across multiple tumor types makes sense from our perspective.
So the aha moment for us, well, actually the aha moment came at our parent company. And our parent company is internationally known and recognized for their services of immunogenicity and safety assessments for therapeutics and vaccines. So they've got 25 years of experience working in the field of T-cell activation and immune tolerance. So one of their important discoveries was the presence of these inhibitory sequences in pathogens and self-antigens. So these are those sequences that I mentioned that downregulate the immune response, which are very important components during the development of therapeutics and vaccines. So almost 8 years ago, our founders were contacted by several cancer patient advocates who were interested and very interested in using our technology to design personalized cancer treatments that would elicit this robust response without the inhibitory effect. So as the requests continued to pour in, the founders realized that there was indeed a need and a business opportunity for our parent company to enter the oncology space. So building on their 20 years of therapeutic design experience, they created Answer, which is this novel bioinformatics platform to design these neoantigen-based therapies. And they spun out EpiVax Therapeutics that would focus in bringing these therapies to the market.
Very good. I now, because a couple of you have raised it indirectly, cancer cells have a multiplicity of mutations. It's part of just what happens with cancer, and it's creates a real challenge in the treatment by the time cancers get to being malignant with a lot of mutation. And each of you have talked about different applications and combination. Len, you haven't talked about it. I don't know if there's a potential risk of some cancer cells developing some resistance to hyperthermia. But could each of you talk a little bit about how the mutation of cancer cells affects the efficacy of your treatments? And let's go with Nicole, Len, and Jeremy.
Okay. So we address the issue by developing therapies that are very specific to the individual patient's tumor, and I think that makes a difference. And we also target multiple mutations at the same time, and I think that we just don't give the cancer a chance to escape immunity at this point.
So that's a great question, Mark, and it's something that has kept us up at night for a while. It's an important point, but we don't have much data on it. And the reason we don't have much data on it is because there really are no what we would call gentle hyperthermia technologies out there. So there's really not much in the way of human data. Initially, our suspicion was that because this is not a chemical entity or a biological, that there could not be resistance developing to it. On the other hand, you see in hot springs and Yellowstone bacteria that have adapted to 100 and something degrees Celsius, right? So it is conceivable that some kind of resistance could develop. The good news, I would say, is that in animal studies, we've seen excellent results with one injection and one treatment with infrared light. So we're not going to be going back to the clinic clearly every week for a year or something like that with humans.
So Mark, from an epigenetic viewpoint, this to me is the promise that epigenetic offers, because it's got that broad applicability across a lot of tumor types, as it's not restricted to one mutational change. We kind of sit above that across multiple gene types, and those those chemical modifications aren't restricted to one type. Yes, they occur in some, but it's not restricted. So this is to us the promise of epigenetics, that we can traverse these mutations in a broad number of solid tissue and hematological malignancies. And that's what we're working hard. And that's why our phase 1 design is an all-comers, to really try and see where the best signals come from. And that, I think, is consistent with my comments.
Okay, so now let's move on to a personal fear factor that people have, that cancer comes and, gee, it's much better if you get it early. And obviously everyone wants to try and catch it early, but relative to the various treatments, relative to the efficacy of other treatments, how important is early intervention with each of your technologies, and how important is the speed of reaction for your particular technology? And let's go with Jeremy, Nicole, and Len.
Yeah, Mark, look, the correct answer is the earlier the better. I think that is fundamentally true. And at EpiAxis, we're wanting to get to that point after we've established the fact that we can treat cancer as a chronic disease. And we believe once we've done that, then low-dose maintenance therapy as monotherapy is important to maintain those cells in a permanent state of quiescence. So when you think about that, we should be applying that right on day one for high-risk patients and patients that have got a primary diagnosis. Why wait for those primary tumors to result in a secondary or metastatic disease? So we believe that this therapy and this type of strategy ultimately is best placed as a prevention style rather than waiting afterwards and then trying to deal with the complexities of having multi-organs involved and an immune system that's kind of been derailed, if you like, as a consequence. So we, even though from a regulatory perspective, it's faster to treat acute disease, so chronic disease, we really want to position it much earlier on in the long run. But that takes a lot of time and money, but that's what we're seeking to do ultimately.
And I'm going to interject here because, Nicole, you talked about getting to the cancers before the immune system had been compromised as well. I mean, that was one of your very specific points, I believe. Now I'll let you carry on.
Yeah, so this is for us, that means a couple of things. It's not just the immune system getting in the way. We are also looking at what we do is activate T cells. So we are an immune therapy company. We want your body to attack the cancer. So we are helping the body attack the cancer. And in this case, having the T cells present is a big deal. So if a patient's receiving several other treatments in advance, you reach what's called the T cell exhaustion stage. And at this point, if you have nothing to activate, your therapy is not going to be very successful. So we want to be in there, close to be in that first line of treatment. Hence, we have shrunk this timeline to 4 weeks, getting in right after the biopsy, giving the patients a real chance here to benefit from this. So that is for us, a very important part of what we do and how we've designed the therapy to be given.
So early detection, of course, is incredibly important. There's no question about that. The good news for colorectal cancer is that if you look even over the last 5-10 years, it is being diagnosed initially at earlier and earlier stages. And that's because of tests like Cologuard, for example, which are being more widely used. So that's good news for everybody, no matter what is out there. One of the challenges in the field is that the standard of care is to use radiation therapy and systemic drug therapy in large part to delay the need for surgery as much as possible. Those are both have a degree of invasiveness, they have side effects, they're costly, and so on, not entirely desirable. Where I think we can really add into that mix, perhaps replace it to a degree in the future, is that we have excellent safety profile, integrates easily into clinical workflow, minimally invasive, very easy technology to use. And so there are really relatively few barriers to using it early and aggressively to say, okay, we have If it's stage 0, you'll go in and you'll have a snare operation done on a polyp. But once you start getting into stage 1, stage 2, you're still not metastatic. You're not even really regional yet, fully regional. An excellent opportunity to do more aggressive treatment at that stage and further delay at least, or perhaps eliminate progression into later stages.
So each of you have actually referenced or walked a little bit into my penultimate question, which relates to cancer treatment having established and very detailed protocols. To a degree, each of you will be disrupting this. Is there going to be a challenge in terms of getting whole groups of people to change or educating them? Or is this something that you feel will work through? So let's go with Len, Jeremy, and Nicole.
No question that it will be a challenge. We've spent a lot of time over the past several years interviewing physicians, patients, patient advocate groups, and so on in a number of different areas. There are a number of cancers we can work with, I think ultimately very successfully. But one of the reasons we targeted colorectal cancer is that we saw very, very enthusiastic responses from basically all of the constituents we spoke to there. And that's in part because there's really not a whole lot that's very effective for it right now. And so I do think it's important. I think integration into clinical workflow, minimal invasiveness are incredibly important. The first for physicians and the second for patients. And increasingly, affordability is important too. And I think we can address all of those things. So yes, we've thought about it a lot. I think we're on the right path, but it will be a lot of work.
Mark, we have perhaps a slightly different view. Yes, it will be challenging, and we should never underestimate that. But we are looking to be an additive and to standard of care. So we're not displacing current therapy. So I think when the clinicians understand that they will use their protocol as they've done today and add something on to get additional performance, we are surmising that will be less of a hurdle rather than introducing a new class of compounds which will then displace. So very importantly, this is an and. And for that reason, we are hopeful that adoption of this will be a little easier than some who have got an entirely new approach, which is a displacement strategy. We're an and that's, I think, a really important point.
I will agree with a lot of that, that Chris just said. But for us, I think that the safety of the product, knowing that it's going to have less side effects than the flu shot, is really easy and convincing fact for most patients and physicians. For the pharma group, I think that it's important to show that these customized therapies can be easily and quickly manufactured, and that these new therapies are not going to be too expensive for the patient and for the insurance companies and so on. I think that we are probably, because we are also not potentially looking to replace the treatment rather than prevent having to get further treatments, I guess it would be more of the case for us, because we plan to go into patients well in advance of the current treatment plan.
Very good. And because we at CSE and Tech Tuesday love to see people succeeding, let's close by having each of you tell us about the next milestone on your road to progress, because we've really enjoyed this session. Nicole, Len, and Jeremy, thank you.
I think that our next milestone is going to be a really important one. We are looking to file the IND and dose our first patient. I think this is going to give the opportunity to this product to really show what it can do. I think we're all very excited here to see this in the clinic. So a big push for us is filing the IND.
Fairly similar for us too. When you don't have small animal data, everyone says you have to get small animal data, of course. The minute you get small animal data, everybody says, well, we're not trying to cure mice or rats. You need large animal data. So our next big milestone is to get those large animal data and show that we're seeing the same results there. And there are also things you can do in large animals that are really impossible or difficult in small animals, such as using endoscopes and getting biomarker data and so on. And so that's a big milestone for us. Once we have those data, I think we'll be well on the way to the clinic.
Yeah. So, Mark, a very similar comment to Nicole and Len in many respects. Ours is the $12 million challenge. We need 2 to get us the IND and 10 to run our all-comers phase 1. And that's really the key milestones that we here at EpiAxis are working very hard to achieve. And that's what we set out to do. We do want to make a difference and leave a legacy. And that's the purpose behind EpiAxis.
Thank you very much, Nicole Ruggiero, Len Pagliaro, and Jeremy Chrisp.
Thank you to the people who've worked behind the scenes, James Black, Melissa Robertson, and of course, the founder and creator of Tech Tuesday and today's moderator, Mark Francis. Next, a thank you to John Ricci and the network at US Angels. Check them out online. Lastly, thank you to our attendees for taking a slice out of your day to learn and support the companies at and on Tech Tuesday. Go to the company websites to learn more about them and their projects. If you have a lead for them, please do connect. Next Tuesday, we'll be bringing you another company showcase. You won't want to miss it. Follow, click, and like on YouTube, LinkedIn, Facebook, and Instagram. May you have continued success in expanding your business.